Chapter 1: The Immune System. Introduction. The immune system has evolved to protect the body from damage caused by microorganisms like bacteria, fungi, viruses, and parasites. This defensive function is performed by leukocytes, or white blood cells, and a number of accessory cells which are distributed throughout the body but are found particularly in lymphoid organs, including the bone marrow, thymus, spleen, and mucosa-associated lymphoid tissues. Lymphoid organs are strategically placed to protect different areas of the body from infection. Cells migrate between these tissues via the bloodstream and lymphatic system. As they do so, they interact with each other to generate coordinated immune responses aimed at eliminating pathogens or minimizing the damage they cause. Lymphocytes are key cells controlling the immune response. They do so by recognizing molecules produced by pathogens. They can also recognize molecules on the cells of the body, although they do not normally react against the body's own tissues. Molecules organized by lymphocytes are referred to as antigens. Lymphocytes are of two main types: B cells, which produce antibodies, and T cells, which have a number of functions, including 1. Helping B cells to make antibody, 2. Recognizing and destroying cells that have become infected with intracellular pathogens, 3. Activating phagocytes to destroy pathogens that they have taken up, and 4. Regulating the level and quality of the immune response. Lymphocytes recognize foreign material by specific cell surface antigen receptors. To recognize the enormous variety of different molecules, the antigen receptors must be equally diverse. Each lymphocyte makes only one type of antigen receptor, and thus can only recognize a very limited number of antigens. But as the receptors differ from each other clone of cells, the lymphocyte population as a whole can recognize a vast range of different antigens. A third minor population of lymphocytes, or natural killer cells, also contributes to antiviral defenses. Phagocytesis include blood monocytes, macrophages, neutrophils. They can internalize or phagocytose pathogens, antigens, and cell debris and break them down. Antibodies and various other recognition molecules bound to the pathogens facilitate this process. Macrophages can also process and present antigens so that they can be recognized by T cells. Accessory cells include eosinophil and basophil, granulocytes, mast cells, platelets, and antigen-presenting cells. Eosinophils have a role in protection against some parasites. Basophils, mast cells, platelets contain a variety of molecules that mediate inflammation. Antigen-presenting cells are a functional defined group of cells. Both B cells and macrophages can present antigen, but leukocyte dendritic cells are particularly important in presenting antigen to naive T cells, which have not previously encountered their specific antigen. Lymphocytes. Lymphocytes constitute about 20% of the total blood leukocytes. The two major populations of lymphocytes, T cells and B cells, are small lymphocytes responsible for recognizing antigen or antigen fragments. A third population of large granular lymphocytes recognize changes in host cells that may occur when they become infected. Large granular lymphocytes are morphologically defined as defined cells containing large amount of cytoplasm with eosinophilic granules, which constitute 5 to 15% of the blood T cells. Both NK cells and gamma delta T cells have this morphology. T cells are lymphocytes that develop in the thymus. This organ is seeded by lymphocytic stem cells from the bone marrow. During embryonic development, the cell then develop their T-cell antigen receptors and differentiate into the two major peripheral T-cell subsets. The helper T-cells express CD4 and the cytotoxic T-cells express CD8. T-cells can also be differentiated into two populations according to whether they use an alpha-beta TCR2 or a gamma-delta TCR1 antigen receptor. The essential role of T-cells is to recognize antigen associated with cell of the host. Gamma-delta T-cells express gamma-delta form of the T-cell receptor. They constitute lower than 5% of the total T-cells, but they are more common in particular sites, including the gut, skin, and vagina. They branch early from the main thymic developmental pathway and recognize different antigens from alpha-beta T-cells, including carbohydrate and intact proteins. Intra-Epithelial Lymphocytes, a mixed population of cells found in submucosal tissue. 10 to 40% are gamma-delta T-cells with a dendritic appearance. The remainder are mostly CD8 T-cells. Cytotoxic T-cells are capable of destroying virally infected or allogenic cells. Most cytotoxic T-cells express CD8 and recognize antigen associated with MHC class I molecules, which may be expressed on all nucleated cells of the body. Helper T-cells perform a number of functions, including helping B cells to divide, differentiate, and secrete antibody, activating macrophages to destroy pathogens that they have phagocytosed, and recruiting cells to site of inflammation. The functions are carried out by different subsets of T helper cells, which differentiate from a common precursor and can be distinguished by the cytokine which they secrete. The majority of helper T cells express CD4 and recognize antigenic peptides presented on the surface of antigen-presenting cells by MHC class II molecules. T helper 1, T helper 2, and T helper 17 cells are subsets of helper T cells originally identified according to the cytokines they produce. They differentiate from a common precursor, or T helper 0. Differentiations of T helper 1 cells is promoted by interleukin 12, interferon gamma, T helper 2 cells by interleukin 4, and T helper 7 cells by transforming growth factor beta and interleukin 6. Dendritic cells are antigen-presenting cells which are most effective at presenting antigen to naive T cells. T helper 1 cells can recognize antigen presented by mononuclear phagocytes, and they interact with this cell by releasing interferon gamma, which acts as macrophage activation factor. T helper 2 cells release cytokines such as interleukin 4 and interleukin 5, which are required for B cell development into plasma cells. Both T helper 1 and 2 cells can modulate the antibody response by affecting the classes of immunoglobulin producers. T helper 17 cells release cytokines that promote inflammatory response, particularly by acting on neutrophils. Some cell surface markers are preferentially expressed on a subset of the helper T cells. For example, the chemokine receptor CCR5 and CXCR3 are more prevalent on T helper 1 cells, whereas CCR3 and CCR4 are higher on T helper 2 cells. All helper T cells can promote the development and activation of cytotoxic T cells and natural killer cells, which recognize and kill infected target cells. Regulatory T cells are a population of T cells usually identified by the expression of the transcription factor FOXP3 and or high expressions of the interleukin 2 receptor. CD25, they can be either CD4 or CD8 and are important in controlling secondary immune response and inflammations, particularly in the gut. They also limit some autoimmune and hypersensitivity reactions acting by direct cell-cell interaction or by the release of anti-inflammatory cytokines, including interleukin 10, interleukin 35, and TGF-beta. They can also inhibit activations of other T cells by mopping up interleukin 2, which is required for T cell proliferation. B cells are lymphocytes that develop in the fetal liver and subsequently in bone marrow. In birth, they develop in a specialized organ, the bursal or fabrices. Mature B cells express surface immunoglobulin, which acts as the B cell antigen receptor. They are distributed throughout the secondary lymphoid tissues, particularly in the follicles of lymph nodes, spleens, and Peyer's patches. They respond to antigenic stimuli by dividing and differentiating into plasma cells. Plasma cells, or antibody-forming cells, are terminally differentiated B cells with expanded cytoplasm containing arrays of wrapped endoplasmic reticulum, devoted to the synthesis of secreted antibody. Plasma cells are seen in the right part of spleen, the medulla of lymph nodes, the mouth and occasionally in sites of inflammation. B1 and B2 cells are B cell subsets. In adults, most B cells are of the B2 subset. They generate a wide range of antigen receptors, mature in germinal centers, and respond well to T-dependent antigens, and cause stimulation via CD40. The B1 subset was originally distinguished by the phenotype CD5+, CD43, CD23-. B1 cells developed early have a more limited range of receptors than B2 cells, respond to a number of common microbial antigens, and sometimes produce autoantibodies. They are absent from lymph nodes, constitute 5% of splenic B cells, and are important in mucosal immunity. Naive or virgin lymphocytes are cells that have not been encountered their specific antigens. They express high molecular weight variants of the leukocyte common antigen, for example, CDR45RA. Clonal selection describes the way in which lymphocytes are activated. During development, each lymphocyte generates an antigen receptor with a single antigen specificity. If antigen is encountered early, the few clones of lymphocytes that can recognize it are stimulated to divide to provide a large pool of effector and memory cells. This is referred to as clonal selection. Memory cells are populations of long-lived T or B cells with some capacity of cell renewal that have been previously stimulated with antigen and can make accelerated response if they encounter it again. Memory B cells carry IgG or IgA as their antigen receptor, which is of higher affinity than the receptor. On naive cells, memory T cells express CD45RO variants of leukocyte common antigens, as well as increases levels of adhesion molecules LFA3 and VLA4. Immunological memory depends on both the production of memory cells and the increase in the numbers of antigen-specific cells produced in the primary response.